So today I would like to talk to you about, so maybe I should tell you where I work. I work at UC Berkeley at the Division of Environmental Health Sciences, which is part of the School of Public Health. And I'm giving a talk about an application of WikiPathways. So my talk is entitled Using the Key Characteristics of Human Carcinogens in a Data-Driven Approach to Improve Carcinogenicity Prediction and Chemical Prioritization. So first, a short introduction about carcinogenicity evaluations, because I'm sure not all of you are familiar with those. So there's many thousands of compounds present in the environment, and there's many compounds being generated by industry. And information on the toxicology of these chemicals is often minimal or absent. And the International Agency for Research on Cancer, also abbreviated as IARC, which is part of the World Health Organization, is responsible for conducting all the evidence about these chemical exposures, both on the epidemiological as well as the laboratory research level. So IARC has published these evaluations in the form of monographs on their website. And how are the monograph evaluations conducted? So IARC has a group of experts for each one of these chemicals, and they conduct these evaluations using a preamble document, which is more like a guidance document. And they have separate criteria for human, animal, and for mechanistic evidence. And then in the end, they have an overall evaluation of all the different types of data and the evidence that exists for those. So what evidence is considered? So there's cancer data in humans, cancer data in animals, and there's the mechanistic data. And all of them together are used for the overall evaluation. So how is the evidence integrated in the mechanistic evidence integrated in overall conclusions? So there's different levels of evidence. So for example, if we start in the upper right corner, there's, for example, if there's inadequate evidence in humans, but there's sufficient evidence in animals, then a compound is classified as a group 2B. If there is strong evidence that a mechanism is also does not operate in humans, then a compound is degraded to a group 3. If there is limited evidence that a mechanism also behaves in humans, then it's classified as a group 2A. And if there is really sufficient evidence in humans supporting the conclusions that were done in animals, then they're grouped as a group 1 carcinogen. So that's a bit in short how these evaluations are conducted. So where does the mechanistic data come in? So there's different human carcinogens which may operate through distinct mechanisms. Many human carcinogens act via multiple mechanisms. And there's no broadly accepted way of sort of analyzing all these mechanistic data together to support cancer conclusions for hazard evaluation. Hazard identification, sorry. So that's why they invented the 10 key characteristics. These 10 key characteristics were created by Smith et al. In 2016. These are different than the hallmarks of cancer. So it's based on the fact that established human carcinogens commonly exhibit one or more of these key characteristics. And data on these key characteristics can provide evidence for carcinogenicity. They can also help in interpreting the relevance of the importance of findings for cancer in animals and humans. And it's a robust new approach to organize and evaluate mechanistic data related to carcinogenesis. And we were wondering how omics data might fit into this KC framework. So the advantage of a data-driven approach is that it provides scientists with an overall image of what is known about a chemical and a disease they're interested in. It provides a more holistic approach, so it doesn't exclude things like you would have in a hypothesis-driven one. And it allows you to do analysis across species, between in vitro and in vivo experiments and different setups. And it uses existing knowledge, which is of course good, because it saves a lot of time and money in doing research that perhaps has already been done. So it provides the opportunity to be more specific about what type of research will be done. So first of all, of course, to perform a data-driven approach, you need a large database. And in this case, we use the comparative toxicogenomics database. The database, as of August 2018, includes 38 million relationships. These exist of chemical gene interactions, chemical disease, and gene disease interactions. And we mainly focused on the chemical gene and the gene disease interactions. And of course, because we're interested in cancer, we used a neoplasm signature, which represents more than 3,000 curated genes and more than 450 cancer types. And curated genes in this sense implies that these genes have been described as certain biomarkers for the disease or their therapeutic targets. And that's where WikiPathways comes in, of course, because we need a way to connect gene association data with the key characteristics. So that's where we used WikiPathways. So, of course, WikiPathways is a very nicely open source and extensive database with more than 1,500 curated biological pathways from different species. And we used WikiPathways in combination with Cytoscape and a specific app in Cytoscape named Clugo to do the Geneset Enrichment analysis, because it's a nice way of sort of integrating different types of data and performing Geneset Enrichment analysis. So, of course, the linking of the biological pathways to the key characteristics is quite crucial. So this was done by 2 experts and were based on descriptions of the pathway or other information that was available on these pathways. So this was done by experts. Then, of course, you also, if you want to use this model for chemical prioritization, you need a way to sort of indicate that a chemical has a strong association with the key characteristics or not. So that's why we invented the KC score. So it represents the strength of the association that a particular chemical has with the KCs of human carcinogens. It's a way of indicating differences between different chemical compounds. And like I said, it can be helpful in chemical prioritization. It's based on the enrichment of the chemicals within the pathways. And we took 8 categories into account. I will tell you a bit more about that later on. And the maximum score was 8 out of 11. Okay, so this is an overview of all the methods we applied. So on the, I don't have a, maybe I have a pointer. That might be handy. Okay, on the on this corner you can see the type of data we collected. So we took the chemical data, the disease data, and we looked at the overlap. And then the overlapping data we used to continue our analysis with. We actually used 2 different databases, the CTD and the drug signatures database. I'll tell you a bit more about that later on. And then we also continued our analysis by doing the Geneset Enrichment analysis. Then, of course, because we had information about carcinogens, we also wanted to know what non-carcinogens are doing. So we overlaid our data with information we had on non-carcinogenic drugs. And then we selected those pathways and genes that remained. Then we sort of connected those with the key characteristics of carcinogens. Then we, of course, looked at, are there any overlaps between the different carcinogens? Do we see a pattern here? And then, of course, we wanted to know which pathways and genes are affected by the majority of these carcinogens. And then, based on these, we performed the KC score calculation, and we did a validation of our approach using 3 different databases to validate our approach. I will go a bit faster. So these are the different groups of the IARC classifications. And the blue bar are the number of compounds that are present in the IARC database. And then the orange bar represents the number that is present in CTD. So for example, for the group 1 carcinogens, we were able to find 57 out of 120 agents in the CTD, which is quite large. Yeah, so in general, we can say that around 68% of the CTD chemicals cover the IARC classified chemicals. We can also conclude here that we, of course, use these 57 human carcinogens as a training set for our approach. Then, of course, we also needed for our training set, we needed a negative group, and thus we used a non-carcinogen group, and that was based on information we got from Mark Fielden, who actually works at Amgen, and he had information about drugs that were proven to be negative in a range of different assays, so in vitro, in vivo, and even like the label warnings that you can find on medication. So we used the CTD and the drug signatures database to obtain these data. So as I mentioned, the drug signatures database is also a very large database. It includes around 17,000 unique compounds with around 20,000 different gene sets. And they're sort of composed by different external databases. Of course, we needed to link the pathways with the KC, so we overlaid the genes that we found for the carcinogens and the non-carcinogens together. And then, of course, we were interested in the genes that were only expressed by the carcinogens. So these are the 361 pathways we found to be affected by carcinogens and then divided over the different key characteristics. You can see that the 0 represents a very big group, so a lot of pathways are not relevant for the carcinogens. We also had an 11th group, which represents sort of the disease pathway, so these are multiple key characteristics within one group. But what was surprising is that we didn't find any pathways for KC2, which is genotoxic. So we actually generated a new pathway, which is called genotoxicity pathway, to add to our approach, and it's also available on the WikiPathways website. And this is sort of a visual of all the pathways we found, and the numbers here indicate the different KCs. You can see that for some KCs we only find one pathway, and for another KC we find a lot of pathways. Here are the KCs explained. Sorry, I have to go through the slides rather quickly. So of course, we then tried to apply our model using a big data validation. So first of all, we used a set of 73 carcinogenic drugs, which was again obtained from Mark Fielden, who had all this information ready. So we used the CTD and the drug signatures database to do this. Then we did a very large big data validation. So we used the entire CTD database, which comprises around 12,000 different chemicals. And then we also used the correlation engine database to do the same thing, but the advantage of correlation engine is that it contains different biosets, which are represented by data from different species, different cell lines, different dosages, and different exposures. So 2 very large databases to test our model. And what we found was that for 29 carcinogenic drugs, we were able to find enrichment using the 33 pathways and 25 genes we selected. From CTD, we were able to find enrichment for around 5,000 genes, and for the biosets, we found 10,000 biosets representing 1,459 chemicals from the correlation engine. So in total, around 6,000 chemicals that showed enrichment. Then, of course, we calculated a KC score for all these 6,000 chemicals. And then we were trying to prioritize them. So how did we do this? So which database gave the highest score when we compared the two? Then we looked at the highest score, and then we said, okay, this is the final key characteristic score. Then we focused on the agents which gave the same score from both of the databases, and we saw that we were able to prioritize 11 IARC evaluated agents and 15 unevaluated compounds, which is of course very interesting because nothing is known about these compounds when we look at IARC. And this is sort of a representation about the score in the different IR groups and also in the not evaluated compound group. So of course you can see that a lot of compounds in group 1 got a score of 8, which was, as I said, the maximum score. And then it reduces the further down you go, which is of course what you would expect because these are like decreasing in carcinogenicity. But there's also a large group of chemicals that are given a score of 8 in the not evaluated set of compounds. So in conclusion, we can say that with our data-driven approach, we were able to link publicly available gene association data with the key characteristics of human carcinogens. We identified carcinogens with few gene association data, so we identified possible knowledge gaps. We were able to identify carcinogen-specific genes and pathways, and we added a genotoxicity pathway to the WikiPathways collection. And we were able to visualize these pathways and genes into a cytoscape network. We were able to develop a model which allows us to separate agents with a stronger association with the human carcinogens from the ones that have a weak or no association. Then we were also able to use our approach or our model for big data validation in chemical prioritization. So we found around 6,000 agents with enrichment in 3 different databases. And we were able to see 11 IARC agents and 15 unevaluated agents with a maximum KC score. A majority of these agents have already been sort of put on a list as being suspect by, for example, by OIHA, which is represented in the Proposition 65 list, which is of course very important here in California. And we also found a lot of anti-cancer drugs, and it might be that the pathways we identified are also very important in sort of fighting cancer. So that is making sense as well. So on future directions, I don't have time for that anymore, sorry. These are the acknowledgements for the people I would like to thank. So from Berkeley, from NIHS, and from Amgen. And that's it. Sorry.
I suggest we take a bit of time for 1 or 2 questions.
Thank you. Very nice talk. I was wondering if you could, do you think you could pull out potentially from transcriptomic data sets, for example, the TCGA individual patients that might have an increased susceptibility for being the gene expression profile being linked to a specific chemical or group of chemicals based on the gene sets that you've isolated here?
Yeah, so that is what we could do. So we could use it sort of as a signature and then see how well these pathways and genes are enriched in these patients. But then, of course, there's a lot of things that play a role if these people are really susceptible to this exposure. So, yeah, we could do it in a way, but how to sort of use it in humans, I think is a step too far for this moment.
Okay. Chris, one last question.
So really nice, Linda, but in toxicology, I'm always interested in the negative results, right? Mm-hmm. So if you look at all these profiles, how many of the known carcinogens would actually not be found by this mechanism, especially if you wouldn't include them in setting up the profiles in the first place?
Yeah, so there is compounds that had a KC score of 0. But those compounds also had not a lot of data in them in the databases, so that could be a reason why you get a 0. But of course, you need to distinguish the ones that get a low score because they have a lack of data with the ones that have sufficient data, and then also give a score of 0. So that's difficult. But I looked at the ones that are expressed by the majority of carcinogens, so that's more than 60% of the ones that we use in our training set. So.
Yeah, the scary thing in tox is that we don't want another softenol, right?
No, exactly. So, yeah. But it's a start, so.
Okay, thank you, Linda, very much for a really interesting presentation.